Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjun classes. Dear student, with a full energy, we are going to start today's new chapter, that is photosynthesis in higher plant. Dear student, as we know that we all need a food for our day-to-day activities, for energy purposes. And obviously, when we need a food for our energy requirements, ultimately, directly or indirectly, we are dependent on the plants. And these plants actually carry out food synthesis. They store within their body and we use them. That's how we get a particular food. So now let's talk about this photosynthesis in detail. Dear student, if we talk about the photosynthesis in higher plant, we know that the higher plants, which include a gymnosperm and angiosperm specifically, they carry out this basic photosynthesis where they use a light. The carbon dioxide they are also going to use, and in the presence of light, by using the carbon dioxide they are going to synthesize the food and releasing oxygen. So all animals including human being depend on plants for their food, as I told you, directly or indirectly. Have you even wondered from where plants get their food? Then we have to think that how these plants get their food. So green plants, in fact, have to make or rather synthesize the food they need. Okay, they have to synthesize their own food, and all other organisms depend on these plants for their needs or the food requirement. So directly or indirectly, because of this, I am telling you, we are ultimately dependent on the plants. Now, specifically, these green plants carry out photosynthesis. That also we know, and this photosynthesis is considered as a physico-chemical process. So now the question is, why it is considered as a physico-chemical process? Because in this, the physical rays, that is a light energy, getting converted into the chemical energy, that is in the form of ATP, and further this ATP are used in synthesis of glucose. So because of this we call it as a what, a particular physico-chemical process. Why? Because it converts light energy into the ultimately glucose or a starch. Light energy is a physical form and the glucose or a starch is ultimately chemical form. So because of this we call it as a physico-chemical process. Now, by which they use light energy to drive the synthesis of carbohydrate. Obviously we know that they use a light energy, plants use a light energy and synthesize a carbohydrate. Ultimately all living forms on the earth depend on sunlight for energy. All living forms are going to depend on the sunlight for the energy. Okay, and the use of energy from the sunlight by the plants during photosynthesis is the basis of life on earth. It is the basis of life on earth. So photosynthesis is important due to two reasons. Photosynthesis is important due to two reasons. First, it is a primary source of food for all organisms on this earth. Okay, because all organisms directly or indirectly depend on food for the plants, and plants carry out photosynthesis. And this photosynthesis is also responsible for the release of oxygen into the atmosphere, which is important for the aerobic organisms or for the survival of aerobic organisms. So that is also one of the important reasons that release of oxygen into atmosphere is carried out by this photosynthesis. So due to this two reasons, photosynthesis is much important. So what do we know about photosynthesis? What do we know about photosynthesis? That first we have to understand. Before that, dear students, we remember that this photosynthesis is considered as an endergonic process. It is endergonic, or we can say endothermic process. It is endergonic or endothermic process because here sunlight is used as a source of energy and energy is going to be used. Because of this we call it as endothermic process. Then this photosynthesis is particularly a redox process. It is considered as a redox process because in this the oxidation as well as reduction steps are involved. And reduction steps are involved. Okay, so this is the thing for this. Okay, now what do we know about photosynthesis? First, we have to understand that. So let us try to find out what we already know about photosynthesis. First, we have to understand that. So some simple experiment you might have done in earlier classes and have shown that chlorophyll, which is a green pigment of the leaf, light and CO2 are required for photosynthesis to occur. Okay, so this basic things you have understood in your earlier classes. Okay, so you might have carried out the experiment to look for a starch formation in two leaves. This is the simple experiment we carry out. We look for the starch formation in the two leaves, and here there is a variegated leaf or a leaf that are partially covered with black pepper and the one that are exposed to light. Now variegated leaf, if we talk, variegated leaf is the leaf which has a both green and non-green parts. Variegated leaf has a both green and non-green parts. And either we take a leaf which is partially covered with the black pepper, and the leaf which we are going to take here, that is going to be exposed to the light. So when we taste these leaves, either a variegated leaves which has a green and non-green parts for the starch, or the leaf which is covered with partially with the black pepper, it was clear that photosynthesis occurred only in the green parts of the leaf in the presence of light. It shows a photosynthesis in the green parts of the leaves. So if we want to understand this experiment, we have to draw it. Now, photosynthesis occurred only in the green parts of the leaves in presence of light. So if you want to understand this experiment, you can see that suppose here this is a stem and we are taking up a leaf. Now in this leaf, this is a green leaf. On this, we are covering it with the black pepper. So this is the black pepper strip. Black pepper strip. And there are we using some clips to hold, so particularly this one. So here we are using some clips like this. So these clips hold the black paper properly. Then we carry out a starch test, that is iodine test. After starch test, we come to know that this is the stem, okay, and this is the leaf. Now after starch test, this area which was covered by a black pepper, so this area of leaf remain unaffected remain unaffected with iodine, whereas these are the two areas which are not covered by black pepper, that areas of leaf affected with iodine. Affected with iodine. So the areas of the leaf affected with iodine and areas of leaf remain unaffected with iodine. Okay, that are the particular things. Okay, now, so it was shown that this leaf remain unaffected, whereas this leaf get affected. So this gives a positive starch test. Means here the starch was present, and this gives a negative starch test. Means here starch was absent. So the area which was exposed to light and which was not covered by the black pepper, which is a greenish in color, that is giving a positive starch taste. But area which was covered by the black pepper is not it giving a negative starch. It means the green part is involved in the what, particularly photosynthesis. Okay, so it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of light from this experiment. Now after this, another experiment was performed, that is known as half leaf experiment, and that particular experiment was carried out by moles, so it is known as moles half leaf experiment. And this moles half leaf experiment proved that CO2 or carbon dioxide is necessary for photosynthesis. So the question is how this experiment was done. So see, in this experiment what was done, this is the leaf, that is a green leaf, and which is attached with the plant. This is the particularly a conical flask, or we can take a bottle like this, we can take a funnel like this. Okay, so now this part of a leaf is enclosed in a test tube with or this conical flask which has a wide mouth, and it is half leaf was inserted inside and inserted here, and the half leaf was remain outside, and here it was properly closed with the split cork, that only the particularly leaf is inserted inside, other any material from outside cannot enter. Such a way we adjust this split cork. So this is the half part which is present outside, this is the half part which is present inside of the leaf. Now here in this conical flask, potassium hydroxide pellet was taken, that is a KOH was taken to absorb the carbon dioxide. So either we can take a KOH solution, or we can take a KOH soaked cotton. We can take a KOH solution or KOH soaked cotton. Okay, now this KOH soaked cotton will absorb all the CO2 from this part. Okay, that is the leaf inserted here. So it exposed the light for the 8 hours, and leaf is taken out and tested for the starch. Now the leaf which was particularly exposed to the sunlight, that is a P region, gives a positive starch test. Means there was a presence of a starch, but this R region gives a negative starch test. Why? Because CO2 was absorbed here. Same way you can perform this experiment like this also. Now, so here in this experiment, a part of the leaf is enclosed in the test tube containing, either we can take a test tube on the conical flask containing a KOH soaked cotton which absorbs CO2, while the other half is exposed to the air. The setup is then placed in light for some time. On testing for the starch later in the two half of the leaf, you must have found that the enclosed part of the leaf tested positive, means which was the particularly exposed part was tested positive for a starch, while the portion of the was that was in the test tube or in the conical flask that tested negative. This showed that CO2 is required for the photosynthesis. Okay, so this shows the CO2 is required for the photosynthesis, that was proved by this half leaf experiment. Now after this, we talk about some early experiment which was done to prove that what are essential things for the photosynthesis and how they are carried out, which scientists involved in this. So when we talk about early experiment of this photosynthesis, then the Joseph Priestley was the first scientist to carry out this experiment. Actually, father of plant physiology is Stephen Hales, who credited the discovery of the photosynthesis. Okay, so first point here, you should also know that Stephen Hales credited for discovery of photosynthesis, credited for discovery of photosynthesis, and he is known as father of plant physiology. And in this, the first scientist who was involved was Joseph Priestley. Now, what Joseph Priestley did? So, in 1770, he performed a series of experiments that revealed the essential role of air in the growth of green plants. And in that, what he did, he taken a one bell jar, and in bell jar, he taken initially a candle. Then candle what stopped burning, okay, or get extinguished as it is covered by the bell jar. So in first setup, the candle was kept covered by the bell jar, candle extinguished or stopped burning. In the another setup, you can see here, he taken a mouse and he taken a candle, burning candle. Then they covered it with the bell jar. When the candle and the live mice, burning candle and live mice covered by the bell jar, after a certain period of time, the mice died. Mice died and candle stopped burning, means candle extinguished. Whereas in this setup, you can see the along with the mice and the burning candle, one potted plant was taken. And when the potted plant was inserted here, that was specifically a mint plant, it was observed that after a certain period of time, observed that candle is continue to burn and mice is alive. Mice is alive and candle continue to burn. Like this, the particularly this thing is going on. Here. So from this experiment, he proved that this mint plant is releasing the oxygen that is ultimately helping this burning candle and the mice to remain alive. So from this experiment, Priestley discovered oxygen in 1774, and Priestley observed that candle burning in a closed space, a bell jar, soon get extinguished or stopped burning. Similarly, mouse would soon suffocate in a closed space. He concluded that both the burning candle and the animal, that is my mouse, that breathe air somehow damage the air. They somehow breathe air and somehow they damage the air because they are releasing CO2. So, but when he placed the mint plant in the same bell jar and he found that the mouse stayed alive and candle continued to burn. So Priestley hypothesized as follows that plant restore the air whatever breathing animals and burning candle remove. Now we know that what the burning candle and breathing animal remove, they remove CO2. That we know now, but that time he was not able to know about this particular thing. So he told it that he described it that the plant restored the air, whatever the breathing animal and burning candle remove. So as a nowadays we understand what they have removed. They have removed CO2 and that used by the particular mint plant, and mint plant released oxygen that ultimately helped this particular one to survive. Because of this, it led to the discovery of oxygen released in the photosynthesis. Now after this, the second scientist who came for this experiment was the Zen Ingenhousz. In 1730 to 1799, he was it is a lifespan of the life period of this Zen Ingenhousz, and he used a similar setup and proved this particular thing. So he showed that he proved. He proved that sunlight is essential to the plant process. Sunlight is essential to the plant process that somehow purify the air. Somehow purify the air, followed by a burning candles or a breathing animals. Okay. He showed that sunlight is essential to the plant process that somehow purifies the air, followed by burning candles or the breathing animals. Okay. So sunlight is essential and that somehow purifies the air, that was proved by this ingenious by using the similar setup that was used by the Priestley by placing it once in the dark and once in the light. He showed that sunlight is essential for the plant process that somehow purify the air followed by the burning candle. So ingenious in another elegant experiment with an aquatic plant showed that bright sunlight, small bubbles were formed around the green parts, while in the dark they did not. So how they done? Just they have taken one particular plant, potato plant, and covered this potato plant with the bell jar and given a light. So in the presence of light, small bubbles were formed over the green parts, whereas whereas the when in the absence of light, these bubbles were not formed. So means in the presence of light they release oxygen. So these bubbles are of oxygen. Later on it was understood. So ultimately he proved that it is the only green parts of the plant that could release oxygen in the presence of sunlight. So later he identified that these bubbles are of oxygen, and hence he showed that it is the only green parts of the plant that could release oxygen. So sunlight is essential for photosynthesis. That was proved by Jennings. That you should remember. Now, after this, the third scientist who was contributed in the discovery of a process of photosynthesis, that was the Julius von Such. In 1854, Julian von Such provided evidence for the production of glucose during the growth of the plant. Okay, production of glucose. And we know that glucose is usually stored as a starch. So, Such later studied that the green substance in plant, that is a chlorophyll, that is in the green substance in the plant, that is chlorophyll as we know it now, is placed is located in special bodies within the plant, and that is called a chloroplast, okay, within the plant cells. So Such later studied that they showed that the green substance, that is a chlorophyll in the plant, is located in special bodies, that is later called a chloroplast within the plant cells, and this chlorophyll are responsible for the photosynthesis that he showed. So here we know that the glucose which is getting synthesized, that is, we know that it is chemically reactive and soluble. Then sucrose, that is chemically inactive and soluble. And starch is chemically inactive and insoluble. So glucose is chemically reactive, it is soluble. Sucrose is chemically inactive, in spite of that it is soluble, but chemically inactive is a starch and it is a insoluble because it is just storage form. So by doing this thing, they showed that green substance in the green plant is responsible for the photosynthesis. So how this the Julius Swan such carried out experiment, and he proved that green substance chlorophyll are responsible for the photosynthesis. So he found that the green parts in the plants is where glucose is made, and that the glucose is usually stored as a starch. Okay. Now, later, if we talk, the Cornelius von Neil came. And if we talk about Cornelius von Neil, he was a microbiologist. The microbiologist based on, and he worked on the purple and green sulfur bacteria. Now, the Cornelius von Neil, a microbiologist, based on his studies of purple and green bacteria, demonstrated that photosynthesis is essentially a light-dependent reaction. Photosynthesis is essentially a light-dependent reaction in which the hydrogen from a suitable oxidizable compound reduces the carbon dioxide to carbohydrate. Means what is happening here? He used purple and green sulfur bacteria, and what he did? He converted or reduced the carbon dioxide into the carbohydrate, and where the suitable oxidizable compound is needed for this reduction to the carbohydrate to or carbon dioxide to the carbohydrate. So this is the reaction where if we are using a sulfur bacteria, suppose we are using a H2S, then if in reaction we use a H2S and react with CO2 in the presence of light, it is going to release the sulfur, then CH2O plus H2O. So sulfur is getting released when there is a hydrogen sulfide. Okay, so hydrogen sulfide when react with CO2 it will form particular sulfur, and CH2O and water is getting released. Okay, so here the H2S if used in place of water, then sulfur is getting released in this purple and green sulfur bacteria. If we use water, then the oxygen is going to release. So here in green plants, water is a hydrogen donor and is oxidized to H2O in some organism. Oxidized to O2 in some organism, it do not release oxygen during the photosynthesis. So when there is the purple sulfur bacteria or green sulfur bacteria, at that time you can see there is no oxygen evolution because water is not used as hydrogen donor. So while H2S as used as a hydrogen donor for a purple and green sulfur bacteria, sulfur is evolved. So sulfur is released. So Neil concluded that oxygen evolved by the green plants comes from the water and not from the carbon dioxide. Cornelius von Neil concluded that oxygen evolved by the green plants comes from water and not from carbon dioxide. So this is the ultimate reaction that 6CO2 when react with the neutral water molecule, the glucose is formed and 6H2 and 6 oxygen molecules are released. Okay, such a way the process is carried out. Where glucose represents C6H12O6 represents the glucose and the oxygen released is from water. This was proved using radioactive isotope also. So this is what particularly the Cornelius von Neil proved, that oxygen so here this is the thing. Now the next thing we are going to talk about that the glucose represents there is 6H12 represents glucose and oxygen released is from water. This was proved using the radioactive technique, that is a O18. Okay, so now this was done by Ruben, Kamen and Hassan. Ruben, Kamen and Hassan. Okay, so this was proved by Ruben, Hassan and Kamen. Okay, now Ruben, Hassan and Kamen, they proved that oxygen evolved by the green plants. So Ruben, Kamen, they proved that oxygen evolved by the green plants come from water and not from CO2 by using this radio isotope technique where O18 radioisotope oxygen were used. Now in this, what Ruben Kamen did, he taken a 6 CO2 in which oxygen is natural oxygen, that is O is 16, plus when they used H2O in this, oxygen is O18 labeled. Now in the presence of sunlight and pigments, C6H12O6 and O16 was used, plus 6H2O plus 6O2 were used, and in this O was O16 and O18 oxygen also was of O18. So C6H12O6 in this oxygen of 16 and 6H2O where the oxygen of 16 and oxygen of 18 were used. So this is the thing where Ruben, Hasid and Kamen worked, and when H2O of O18 were used, oxygen also of O18 was released. Means oxygen comes from water, not from CO2. That was proved by this particular experiment. Apart from Ruben Kamen, the next scientist was Robert Hill and Bendal. Now Robert Hill also done one experiment. He done a detailed study of light reaction by using an isolated chloroplast of Stellaria. So they used isolated chloroplast of Stellaria plant, and in a beaker they have taken a chloroplast of this particular Stellaria media. So this is isolated chloroplast. Okay. Now in this, this isolated chloroplast was used. Then apart from this, reduced hydrogen acceptor were used. And it is a CO2 free medium. So CO2 free medium, CO2 is absent, and in place of CO2 the reduced hydrogen acceptor was provided, and this hydrogen acceptors are ferricyanide, then others like chromate or DCPIP can be used. DCPIP is dichloro. So dichlorophenol indophenol. So that all can be act as a hydrogen acceptor in place of CO2. So CO2 will accept this, this CO2 is not that, so they will accept hydrogen and glucose synthesis will occur. And it was observed that when light was given to this chloroplast, in the absence of CO2 also, the oxygen evolved. This means if CO2 is not there, but water is there, and then also in the presence of absence of CO2 and presence of water, oxygen is evolving in the presence of light, means the photosynthesis is occurring. Okay, so that's this Robert Hill also proved that oxygen evolved from water and not from CO2 by using this chloroplast of stellaria media. Okay. Now, after Robert Hill, the next scientist who worked here, that was T.W. Engelmann. Now, T.W. Engelmann was one scientist who used a prism to split the light into its spectral components and illuminated a green algae, that is cladophora, and placed in a suspension of aerobic bacteria. So what they have taken? They have taken a prism, they have provided a light and splitted it into the different colors, okay, and given it to the suspension of the cladophora and aerobic bacteria. So bacteria were found to associated with the cladophora algae by using bacteria used here to detect the sites of oxygen evolution, and they found attached to the this particular aerobic bacteria algae. So the bacteria accumulated mainly in the regions of blue and red light of the split spectrum. And the first action spectrum of photosynthesis was described, and it resembles the absorption spectrum of the chlorophyll A and the chlorophyll B. So from this, the blue and red light are important. That was proved here by using this cladophora, suspension of cladophora and aerobic bacteria. And by the middle of the 19th century, the key feature of the plant photosynthesis are known, namely that plant could use light energy to make the carbohydrate from the CO2 and the water. Okay, so that was the discovery with respect to the what particularly we can say the T.W. Engelmann. Now, let's discuss some of the MCQ. The end product of photosynthesis, obviously, it is starch. Who reported that the role of air in the growth of the plants? That is Priestley. And who experimentally proved that sunlight is essential for photosynthesis? That is Ingenhousz. Who proved that evidence of glucose production where plants grow? So that was given by such that glucose production when plant grow. Okay, so that is all about the what early experiment and the initial introductory about the photosynthesis. From the next lecture we will be discussing about the where this photosynthesis takes place and how this photosynthesis takes place and how what are the reaction are involved in this. I hope you understood well till this. Thank you everyone.